that was another astounding talk. Several aha moments. Ashok, please. And Larry Steinman, come on up and be a surrogate for Sir John. I know you'll do him proud. We've had just an incredible session here in terms of profound insights that are certainly not. Why don't you move over here? Oh, okay. Yeah, that's good. Okay, good. Well, where to begin? Larry, a reaction?
So, Ashok, there are a number of cancers, as was shown, where obesity plays a risk, and you have these wonderful technologies and ideas. How can you drive that connection? And one of the cancers that, because I'm always looking for the outliers, that does not seem to be associated with obesity, there's a statue of Bobby Moore, most famous British soccer player. He had testicular cancer, but obesity doesn't affect that one. So is that going to teach us something?
Larry, those are good questions. Let me take the second one first. You may have noticed that I emphasize the importance of tissue context. And that's because I feel it's often overlooked when people discuss the relationship between obesity and cancer. The fact, as you pointed out, that not all tissues are predisposed to cancer in people who are overweight, have metabolopathies, or are obese, to me indicates that there are probably some types of cancer whose pathogenic mechanisms are independent of the systemic metabolism, cellular homeostasis, chronic inflammation triad. It makes sense to me that a rapidly dividing tissue, such as the testicular germ cell, in which there is also the need to regulate the transition between meiotic reduction and mitotic cell divisions, you perhaps don't need to invoke systemic metabolism in order to see where there might be ample potential for the generation of genome instability. But to me, what's far more intriguing is the differences amongst the tissues that are predisposed to cancer. I pointed in my slide to the difference in risk. For example, endometrial cancer is described as having perhaps a 7 to 10-fold risk. Many of the other visceral organ cancers, pancreas, esophagus, etc., about a 2 to 3-fold risk. Now, the point to emphasize there is there are a number of factors that could be playing a role. For example, in esophageal cancer, it strikes me that the incidence of hiatal hernia in obese people, which predisposes to acid reflux, which is a known risk factor, could be something that doesn't require a deep mechanistic explanation. It's an anatomical thing. Whereas for the other visceral organs, it's pretty clear, I think, that inflammation plays roles, but different roles in the way that you get cancer in those tissues. So I think this is a really important point that bears emphasis, and I hope you'll forgive me for belaboring it because I think it's often overlooked. Second point, how can we use the technologies like protein interference and the like that we have developed to address this issue? We've started with our colleagues in Cambridge at the Metabolic Diseases Unit, as well as at the Mitochondrial Biology Unit. So we believe we can look at phenotypes that people have already identified as being, as marking key changes that are altered in inflammation or obesity or both, and begin to identify underlying pathways. And through protein interference, find targetable rate-limiting steps in those pathways. The complexity here, as opposed, for example, to genome instability, which is largely a cell-autonomous phenomenon, is that most of these links are not intrinsic to a single cell. but rely on cooperation between different cell types, immune system, adipocytes, epithelial cells, and so on. That's a challenge. But we are in the process of developing assays, systems, which can actually cope with multiple cell types interacting and still read out phenotype. So we are cautiously optimistic, is that the right phrase, that we can tackle these technological problems and identify rate-limiting steps.
Thank you.
Eric, I love the picture of the silos. That really gets right at what we're trying to approach. The cancer silo is one that has largely been outside the aging biology domain, as best I can tell. And I'm wondering if you have gained any insights listening to Ashok that you might pursue with him.
It was quite an interesting talk, fascinating. It has been in people's horizon, and there is actually quite strong evidence that many of the pathways that control aging also affect cancer. And I'll give the example of Laron dwarfs. These are humans that carry mutation, growth hormone signaling. Growth hormone is one of the major pathways. Again, it goes via IGF-1 and eventually leads to cell growth. It's one of the major key driver of aging, but also of cancer. And these patients, the Laron dwarfs, actually appear to be protected. There's also evidence, for example, calorie restriction protects against many different cancers. There's evidence that metformin actually protects against cancer. So there are multiple links, both genetic, pharmacological, that already have established this relationship. Obviously, there's a whole other level of complexity that we do not appreciate. And I think when I hear Ashok talking about this role of the context of the tissue is really a key aspect that we have not addressed. But the link is clearly established. All of the work that will need to be done is to see, for example, how can we actually translate this into cancer preventative therapies.
We can take a question from the audience, if there is one, and if not, we'll have plenty. Yes, sir.
Thank you. My name is Kees Kostanje. I'm working for Astellas in the Netherlands. I was triggered by the beautiful lecture of Professor Venkitaraman and revealing new technology to identify different pathways, but pathways that could affect the kind of reverse degradation of other elements. And I was triggered by the fact that, yes, this is meaningful, but would like to give you also a kind of suggestion here, because compartmentalization in the body is very vital to a lot of compounds, like, for instance, formaldehyde, CO, NO, and we know that what these are doing, for instance, also in certain conditions with immune system constituents. And so I think we also need things like quantitative systems pharmacology to understand where a certain compound needs to be at a certain time in order to be effective. I've been working a bit in that area, so I know also a very nice example where you can change one compound moving from one silo to the other silo.
I think you've raised a very important issue in relation to aldehydes in general, and I didn't have time to go into some of the complexities of the system, but I think you're absolutely right that these are very reactive compounds. They're not just coming in from the environment, but they're also importantly being produced by systemic metabolism and cellular metabolism. Because they're very reactive, there are a number of different cellular systems that mop them up and therefore compartmentalize them in the way that you say. So I think understanding, if you will, the systems pharmacology of this type of thing is something which is very high on our horizon at the moment. The difficulty is, again, you anticipated this, assay systems. The existing assay systems for a lot of these metabolites, not just the aldehydes, some of them are relatively straightforward to measure. There are well-developed, robust assays, but we're increasingly finding that for many of these reactive compounds, there is just no assay available that can measure transient or cumulative exposure. And I think assay development for not just aldehydes, but a number of different metabolites is, again, high on our horizon for the reason that Larry alluded to. These are phenotypes that I think we can then begin to use protein interference and other technologies to understand how they're generated, where they're generated.
Larry, if we don't have a question from the audience, we'll take one from you.
Well, the aldehyde is a famous metabolite of ethanol, and ethanol was connected through red wine to sirtuins. So this has led a lot of people, including pharma, to invest very heavily. Where is that going? What are the implications?
I think Eric may be better poised to answer the resveratrol question. I can certainly comment on the mutagenic versus the protective potential of alcohol. I'm not sure that it's a good balance, personally.
So the resveratrol story is, how do I put this? It has not panned out in terms of, there's too little resveratrol in red wine to actually activate sirtuins. It's been verified that resveratrol is indeed an allosteric activator of sirtuins. There's a structure now, there are mutations. So the controversy in terms of whether resveratrol is an activator of sirtuins has been cleared. What has not panned out is the idea that resveratrol could be used as a therapy against aging. And I think it partly reflects the complexity that it only activates one sirtuin, SIRT1. There are 7 of them, and that you probably need to activate them in a much more complex manner than what resveratrol can do.
Well, there was a heavy dose of wishful thinking in that hypothesis, and yet it does seem to be a phenomenon that drinking wine, maybe red wine more than white wine, is beneficial. It almost certainly is not resveratrol, but what would it be? It's probably not the 2-carbon fragment necessarily. What else might it be in a bottle of red wine?
I'll just add maybe one hypothesis, which is only a hypothesis. I think there's growing evidence that a lot of the compounds now that increase lifespan all induce what we call hormetic responses. So many of the compounds that increase lifespan actually are mildly toxic by themselves. And so they might be actually activating protective mechanism. There's even a theory about why vegetables might actually be healthy in terms of lifespan. One of the theories that I think I find satisfying is the idea that most vegetables actually contain mild toxins that have evolved for the purpose of protecting the vegetables from being eaten by prey. And a good example is resveratrol. It's actually secreted as a mild toxin. It's a poison for mitochondria. And we know that many of these mild stresses actually induce protective responses that tend to increase lifespan.
Fascinating.
And there's a wonderful counterexample, Guam. So Guam used to have a fairly low incidence of obesity, but they had this ALS, Parkinson, dementia complex. And now, since people stopped eating cycat, that went away, and it's been replaced by McDonald's-induced obesity. I think there's a lot of sociological tales about that trade-off from going from the ancient diet. There's also, talking about ancient diets, in the Native American population in the Southwest, there's an intense level of obesity. And that, again, may have had evolutionary advantages, feast and famine, And now it's crippling because of you don't have to move, you just drive to McDonald's and eat.
I think it's an excellent point. I think the example of Guam is really one that I think for me inspires sort of humbleness in terms of the task that is at hand, because the curve I showed you in terms of food intake and lifespan is so-called hormetic curves. You see this, for example, paraquat is a really good example. Paraquat, which is a mitochondrial toxin, creates intense oxidative stress. At low doses, increased lifespan for partly the same reason that I've said about vegetables. However, if you cross that threshold, it induces actually accelerated death by the same reason. So these hormetic dose-response curves, we see them for many, many compounds. We see them for food. And I think they signify that it's going to be very hard to find, even if we find a drug that increases lifespan, to find a sweet spot where you increase lifespan and don't actually do the opposite. So,
there's another layer of complexity, and hopefully in the afternoon, Thomas Olson can speak about it. But smoking, generally bad, cancer. It does reduce obesity. However, the nicotine, the natural product that causes the addiction, stimulates an alpha-7 acetylcholine receptor that has regulatory effects, and indeed, for certain diseases, and we should let Thomas talk about it in the afternoon, but Parkinson, it's protective inflammatory bowel disease. Nicotine is quite protective. So parsing out, inhaling the nicotine, the smoke is bad, but the nicotine itself, just aside from its addictive properties, has benefit.
We'll give the last question of the morning to Philip Home.
That's very kind. Listening to that conversation, I was just thinking to myself that, all foods are toxic. I mean, the reason we control glucose so tightly, we can debate forever, but ultimately, if you do the calculation, we actually waste 30% of our calorie intake of sugars, storing glucose away so that it doesn't rise to a high level in the concentration. And of course, if you throw glucose on a fire, you can see why. It's a reactive substance, it's a fuel. That's what my brain is burning at the moment. Amino acids, of course, I don't need to point out, highly toxic substances for perhaps slightly different reasons. Alcohol we've mentioned. That brings me to fatty acids in the basis of my question. My blood is full of albumin, and one of its prime reasons for being there actually is to transport fatty acids between my tissues, because fatty acids are highly toxic substances. And I'm wondering, Ashok, listening to you, whether you talked about aldehydes, but whether actually fatty acids and their huge diversity of metabolites couldn't be playing a similar role in your proteomics.
I'm glad you raised that point. I very specifically alluded to it because I do feel that the mechanism that we have described may be essentially opening a window into potential direct effects of smaller molecule metabolites, including fatty acids. I suspect that they are going to have direct effects on gene expression, direct effects on the stability, the chemical stability of DNA, such as we've described for the aldehydes, which are really not explored yet. And it makes a lot of sense to me from what Eric has said from the discussion before, that some of these are responsible both for reactions involved in aging as well as potential predisposition to disease.
And
Eric,
we can't forget that fatty acids drive, whether we want to eat or not, via octanylation of ghrelin. Complex.
And we have omega-3 fatty acids, Eric, and what do we do about that? They seem to be good in many ways.
Well, I don't know about fatty acid, but I just want to add one more thing about glucose. Someone talked about Tony Serami earlier, who discovered the role of TNF and as a cachectin. Tony also published a series of papers on advanced glycosylation end products, which are direct chemical reaction products of glucose. And I think these, there were some clinical trials of drugs that failed, but the field is undergoing a whole resurgence, really re-examining the role of glucose as a direct byproduct. That could link, again, toxicity of diabetes and with pathology.
While we're in praise of Tony Cerami, he made one other massive contribution to the subjects we're talking about today. He invented the A1C test. And he did that research at Rockefeller. One of his study subjects was his mother, who was in the first New England Journal paper.
Well, with that fascinating discussion, we'll bring things to a close and we'll look forward to the next session after lunch. And I hope that you'll intermingle during lunch and share ideas. This has been a wonderful
And
morning.
When we reach 80% of what we think we should eat, we should stop.
Yeah, that's a good one.